Thanks very much for the opportunity to present our research on virotoxigenic E. coli in private wells. So I didn't have too far to come today. I'm with the University of Galway and I'm in the antimicrobial resistance and microbial ecology group in the discipline of bacteriology, which I realise is a mouthful and quite difficult to... we're also in the Centre for One Health, so we do a lot of research involving the interaction between animal health, human health and environmental health. So I'll be talking today about virotoxigenic E. coli. So first of all, an introduction to what it is. Well, it's a pathogenic strain of E. coli. It's associated with ruminants, so farm animals, primarily cattle and sheep in Ireland, but also pigs. They produce toxins, which are essentially poisons that cause the symptoms of the disease. And it's a food and waterborne type disease. It causes severe gastrointestinal infections, which are characterized by bloody diarrhea, abdominal cramps and vomiting. Importantly, there's a very low infectious dose, so as few as 10 cells of the bacteria are enough to cause disease in a susceptible individual. About a third of those infected require hospitalization to recover. Most cases occur in children under 5 years of age and in the elderly. Not that common, but a very serious complication of the disease is hemolytic uremic syndrome, which is a kidney-associated disease. This occurs mostly in children, and unfortunately it is potentially fatal. So VTEC has a particular seasonality in Ireland, and I suppose it's really linked to the grazing season. There are two main peaks for the two main serogroups of the pathogen. So the 026 serogroup has a peak in summer, in June and July, and the 0157 serogroup of the pathogen has a peak later on in the year in September and October. Other than these two main serogroups, there are also lots and lots of other serogroups of the pathogen which are capable of causing serious infections in humans. As far as the national distribution of VTEC infection, it's more common in rural areas, which you would expect, I suppose, since it's associated with ruminant animals. So the Midwest, the Midlands, the West and the Southeast are the most common areas of the country affected by VTEC infection, and the crude incidence rate in these areas is very high. We are also the top country in Europe for VTEC infection, so we have the highest crude incidence rate of all the countries in Europe for VTEC infection. And we've had this for quite a long time. You can see from the chart on the bottom there, the peak in 2018. Climate really is impacting the occurrence of this particular infection, so extreme weather events lead to an increased rate of VTEC infections. So why Ireland? Why do we have such a problem with VTEC? Well, Ireland can be described as the perfect storm for this pathogen. So we have about 750,000 people who are using groundwater for their daily drinking water source. 500,000 of those are using private household wells, which are unregulated at the household level. What this means is if you have a household well, it's your responsibility to get it tested. It's your responsibility to install a treatment system if it's needed and to maintain that treatment system. And of course, all of this costs money. Groundwater supplies can be highly vulnerable to contamination with pathogenic microorganisms, and in Ireland we have plenty of the sources, so we have over 14 million animals. We've got a lot of rainfall, especially here in Galway, but then in other parts of the country as well. We've got geology that's susceptible, so a lot of the country is karst geology, which there's really little ground cover, and what's on the surface can quite easily get down into the groundwater, into people's wells and into people's bodies essentially through drinking water. So it's important to mention that as well as agriculture, which is the primary source of the pathogen, secondary source is through human feces, obviously through septic tanks, and as Stephen alluded to, if septic tanks aren't looked after properly, this can end up in the groundwater and potentially contaminating the well. Private well use is the top risk factor for VTEC infection in Ireland. So although only 1 in 10 people in Ireland are using a private well, 4 out of every 10 people who are infected with VTEC have been exposed to private well water. So I'm just going to take you through some studies that we've done over the last couple of years looking at contamination of private wells with E. coli and VTEC. So the first is a study that was done actually by my colleague, led by my colleague Dr. Gino Dwyer in UCC. She examined EPA data that had been collected from over 132 water supply wells, both private and public, over a 10-year period. analysed 3,800 samples in total, and she found that 68.2% of water supplies were contaminated with E. coli at least once during that 10-year period, and 13.5% of all of the water samples tested positive for E. coli. So if you're interested in finding out more, the study is published in Science of the Total Environment last year. As you can see from the maps on the right there seems to be more of a problem in more rural areas, the West and the Midwest, and there is a seasonality associated with the contamination rate as well. So moving on then, we wanted to look specifically at viral-cytotoxigenic E. coli, VTEC, detection in wells, and we carried out a study which was funded by the EPA, where we examined 52 private wells in Western Ireland and assessed them for contamination during the peak season, the September-October in 2019, and we were supposed to do it in 2020, but it ended up getting put on hold again until 2021 because of COVID. So what we found is that 38% of wells were positive for E. coli, and there was actually quite a high median CFU of E. coli in the wells, so 9 per 100 mils. VTEC genes were detected in 19% of wells overall. So this indicates that VTEC was present in 19% of wells, and 40% of wells that contained E. coli also contained genes for VTEC. So this is like a really high rate when compared to internationally. As well as that, we found multiple clinically relevant VTEC serogroups in wells that were highly contaminated with E. coli. So if you had a well contaminated with E. coli, it was more likely that there was a few different types of VTEC bacteria there that were capable of causing human infection. So 1/3 of E. coli contaminated wells contained a VTEC gene indicating the presence of VTEC, but also this other gene, EAE, which is a virulence factor that the bug uses to cause more serious types of infections. So those more likely to be associated with that complication that I spoke about, hemolytic uremic syndrome. So this study was published recently in Science of the Total Environment if you're interested. So from these studies, I suppose we were able to identify what the key risk factors were for well contamination. So hydrogeology is very important. So we found that it was much more likely that E. coli would be present in wells in areas where there was extreme or rock karst groundwater vulnerability. So where there was low soil cover and karst geology, much more likely that wells will be contaminated. We also made an association between septic tanks and the presence of E. coli. So a lot of the wells that had E. coli had a septic tank within 100 meters up gradient of the wellhead, and also in areas where there was a greater septic tank density, it was more common to find VTEC contamination of wells. So rainfall is also another big contributor to wells becoming contaminated, and we found that a mean increase in the 30-day mean antecedent rainfall was associated with VTEC contamination of wells. And finally, well design is also the other big factor, I suppose. So the better the wells were constructed, the better they were protected from VTEC. And we found that wells that were shallower were much more likely to be contaminated with VTEC. So what does all this mean for public health? Well, if we think about the numbers, there are about 120,000 private wells in Ireland serving about 500,000 people or so. There's also small group schemes which also use groundwater drinking water sources. From our research, we can estimate that around 30 to 68% of these could be contaminated with E. coli at any given time. That's a lot of wells potentially contaminated. And if we consider that 40% of these potentially have VTEC, it's a big problem. So we know from Health Protection Surveillance Centre data that the crude incidence rate for VTEC in Ireland in 2021 is 17.5 cases per 100,000, but that's in the general population. In people who are using private wells, the crude incidence rate is likely much, much higher than that. Conservatively 5 times higher and maybe as much as 10 times higher than that. So, that's potentially a lot of children that are going to end up in hospital with bloody diarrhea, which is obviously very distressing for families. And it's mainly rural families that are affected. So, it kind of involves all of us. So, I suppose the EPA have reacted to this by putting out a call for a research project that helps us to detect and manage the risk of VTEC in our water environment. So we just started last year this project DERIVE, Detection and Risk Management of VTEC in the Water Environment, and it aims to draw evidence-based recommendations for policy pertaining to detection, monitoring and regulation of VTEC in our water ecosystems and in our drinking water supplies. So we hope that the findings of the project will inform novel risk mitigation and management measures, especially in light of the new Drinking Water Directive, which is more focused on kind of managing the risk at catchment-based level and breaking the pathway from source to receptor. So there's two main technologies that we're developing as part of the DERIVE project. So the first are rapid molecular tests that we can use to detect VTEC presence in wells. So we have two different types of tests that we've developed. We have a quantitative test that can help us understand what is the concentration of VTEC, two of the main serogroups in VTEC in water supplies, whether they're drinking water supplies, but also in natural waters. And then the second type of molecular test, which is pictured here, is a rapid on-site method for detection of VTEC in drinking water supplies. So the idea for this test is that an environmental health officer, for example, can use the test during outbreak investigations to, within 45 minutes, be able to get an indication whether a well is contaminated with VTEC, and if it is, they can send the sample back to the lab for further analysis. The second tool that we're developing is a little bit more complex and takes a lot longer, I suppose, to develop. And we're currently undertaking field studies to generate the data to allow us to develop this tool. So we're hoping to generate a risk-based predictive modeling tool that has a simple graphic user interface that people can use to predict how likely it is that their well will be contaminated under different conditions. So different weather conditions, different climate conditions, different hydrogeology conditions, etc. So currently we're carrying out field studies in one catchment, and we're hoping to also do a second catchment then next year. And we hope that combined that these two technologies will help us to manage the waterborne VTEC risk and mitigate the effects of it on people's health. So I suppose just to take you through what we've done on this DERIVE project so far. So we started out by identifying which study catchments we want to focus on, and to do this we mapped the risk of VTEC infection. So we mapped all of the human cases of VTEC that have occurred in Ireland over the last number of years, and we collated that with information on susceptibility. So where were the density of septic tanks, like Stephen spoke about, where is the rock more susceptible, where are the high concentrations of the source animals, and this is what we came up with. So it allowed us to identify study catchments that we can use, and I mean, we'd love to do the whole country, but we don't have the possibility to do that, so we're choosing two catchments that are representative of the country, and the idea is to develop the model and then test it to see if it fits the whole, so we can scale it up to a national perspective. So the first study catchment that we chose is within the Corrib catchment, and it's the Black River catchment, which is located to the west of Tuam, in between Tuam and Headford. So we've engaged with multiple stakeholders within the catchment to identify different sources that we can sample. So we've been sampling boreholes, private wells, and also group water schemes, and we've been liaising with the National Federation of Group Water Schemes who've helped us with that. We've also been working with LawPro to help us identify stakeholders within the catchment, and with the Teagasc ASAP programme who've helped us to engage with people from the agricultural community. So right about now we've identified all of our sampling sites, and we've already done about 3 samples taken so far. So here's just an indication of some of the sites. We have boreholes, more shallower dug wells, rivers, and this area is mainly a beef and sheep agricultural area. We've also managed to validate our rapid molecular test using river water from the Corrib, which has shown that we are capable of detecting VTEC on site within 45 minutes with this test. So this is the study team. So I'm presenting here today, but obviously most of the work has actually been done by a lot of the people that are here. So as well as the University of Galway, University College Cork, TU Dublin and Teagasc are involved. We also have a key stakeholder group which are helping us to connect and advising us, and they include people from public health, from environmental health, from the Health Protection Surveillance Centre, Noel from the ASAP programme who spoke yesterday, and Barry Dean from National Federation of Group Water Schemes and the local water conservation officers. So I suppose what can we conclude from this work? Well, we know that poor well design is one of the key risk factors for contamination. Our recommendation is to establish more stringent guidelines for well design, boring and maintenance. We know that communication with private well owners is really important, and EPA have done lots of good communication with private well owners. We're hoping that the DERIVE project will help us to understand when we can target communication campaigns a little bit better. So where there are areas of high risk, we think that group schemes might be more effective than private wells, might be easier to manage. So the recommendation is to identify these areas and liaise with local communities in order to set up group schemes which can be treated and routinely monitored. And then we know that the sources are agriculture and septic tanks, although the relative contribution of each of these different sources is currently unknown. We're hoping to find out more about that through the DERIVE project. But it's important to involve all stakeholders here. We all have a stake in this. A lot of the people who are well users are also farmers. They also want to protect their families from this infection too. And like I said earlier, climate has a huge impact on pathogens in the environment. And it's really important, I suppose, that that's taken into consideration when we're trying to develop policy, and that drinking water and private drinking water is taken into account in the National Climate Action Plans. So thanks for your attention. This is my contact information, and I'm really open to any stakeholders that would like to participate in the project, whether you're a private well owner in the Galway area, and we're also planning to do a study in the Limerick area in the future, to get in touch. And if you like this, you might also like another talk that's going on later today at 2 o'clock in the Galway Hotel. My colleague, Professor Derval Morris, is presenting the results of her ARREST project, antimicrobial resistance in the environment.